My name is Melia DeVivo. I'm going to give a presentation on the factors that influence ungulate populations. So Scott did a really nice job of introducing you to some of the terminology and the how ungulate populations grow. So I'll be using a lot of that same terminology throughout this presentation, and also giving you some more context and some specific examples of how this plays out, and how certain factors can potentially influence populations very broadly. So for today's presentation, well, and now my screen doesn't want to advance. Hold on. Oh, there we go. Okay, so I today will focus on five main factors that potentially impact populations, or more specifically population size. So those factors include climate, human activity, predators, resource competition, and parasitism and disease. So these factors can be categorized into two different categories: those that are density independent and those that are density dependent. And so Scott mentioned factors that are limiting. So all of these factors have the potential to limit populations. Some of these factors will potentially regulate populations. So when we talk about regulation, that typically means those factors that are density dependent. So operate on a population and affect them depending on that population's density, as compared to density independent factors that are going to affect population size regardless of the population density. You'll notice that that line of density dependence isn't perfectly encompassing parasitism and disease factors. And so I'll talk here shortly about that, specifically why that factor doesn't fall completely neatly into one or the other category. So for the remainder of the presentation, I'm going to present each topic, and here we go. So the first factor we'll talk about is parasitism and disease. The first thing that I like to tell everyone is that all wildlife are diseased or parasitized in some way. It's just that some of those animals don't actually succumb to that whatever ails them. So just have that in mind, that there really is no wildlife that are absent of some sort of parasite or disease. Generally speaking, again, this factor is a density dependent factor, but there are situations and certain diseases that don't fall very neatly in this density dependent category. And so an example of that are that some diseases don't require direct transmission, so a sick animal infecting a susceptible animal. So some diseases can infect animals through contaminated environments. Some diseases require some sort of vector or an intermediate host to transmit the disease. And then those diseases can kill what we call dead end hosts. So hosts that don't perpetuate the disease cycle. So in these scenarios, the density of the dead end host doesn't actually affect the probability of transmission, or it doesn't increase the infection rate. So on this slide, I have the life cycle or life history of a disease called brainworm. So this disease, you might not have heard of it because it actually doesn't affect populations here in the West. It's primarily a disease that you see in Eastern ungulate populations. So the brainworm actually, it naturally cycles through white-tailed deer populations. And so white-tailed deer species is actually perpetuates the disease. It does have an intermediate host, which the larvae live in snails or slugs. And so when white-tailed deer are foraging and they accidentally consume these snails or slugs and they pick up the larvae develop within its natural host, the white-tailed deer, and the white-tailed deer sheds the larvae in their feces, and then that whole cycle perpetuates. Unfortunately, other ungulates, such as moose, are dead-end hosts. So when they accidentally eat these infected snails or slugs, the parasite actually will kill that host if it's a species like moose. So in this scenario, the density of the moose population does not actually contribute to disease transmission. So this is just one example of how disease isn't necessarily density dependent. So another example of I'll say a not so density dependent disease is hemorrhagic disease. Hemorrhagic disease is the most important viral disease of white-tailed deer in the United States. It's transmitted by a biological vector, and these species are in the genus Culicoides. This viral disease is seasonal, and so outbreaks occur typically late summer, early fall, and it ends with the first frost. And that's because it's really dependent on being transmitted by that biological vector. So it's timed with when the Culicoides midges are out and taking a blood meal from deer. Excuse me. When we do see outbreaks of hemorrhagic disease, we can sometimes get reports of hundreds to even thousands of dead deer on the landscape due to this outbreak. And so outbreaks are truly driven primarily by climate because it is tied to the vector that transmits the virus. Obviously, the prevalence of the vector of the Culicoides midge, and then there's also a component of host population immunity. So in the case of hemorrhagic disease, deer that survive from an infection can actually have an immune response that they can then pass on to their offspring potentially. And so it's not, these outbreaks or this particular disease is not necessarily driven by deer densities. However, overpopulated herds could be potentially in poorer condition. So this is where, as Scott described, so in populations that are really feeling the effects of density dependence and with limited resources, those animals will likely be in poor nutritional condition. And so they'll be less resilient to potential outbreaks of hemorrhagic disease. So there is an indirect effect of populations and population density on how they interact with disease. Also, the high reproduction that we see typically in white-tailed deer populations, as well as this immunity factor, may actually curtail long-term impacts of hemorrhagic disease. Now, this is probably a kind of an outdated, I don't know what you want to call it, thought process potentially, because when it was thought that maybe a hemorrhagic disease, when it was first documented back in the late 1800s, it potentially actually regulated white-tailed deer populations in the Southeast. So you have these overabundant white-tailed deer populations, and then you get an outbreak of hemorrhagic disease. Well, so then you reduce the population density, and then as Scott has previously described, then you have potentially more resources on the landscape. And so then those remaining deer are able to thrive in those populations, because many of their the interest-specific competition was removed because of this disease outbreak. Now, we're more concerned with how frequent these outbreaks are occurring because of climate change. So again, climate plays a pretty big part in outbreaks of hemorrhagic disease. And so with more frequent droughts and conditions that really allow that vector to thrive, we're seeing more frequent outbreaks. And so if these outbreaks are occurring more frequently, potentially the white-tailed deer populations aren't able to rebound like they once were, when outbreaks were only occurring every 5 to 7 years. So this is just an example of how disease some diseases don't perfectly fit into that density dependent category. So let's shift and now talk about a disease that fits better into that density dependent category, although I will say it's not perfect again either. So let's talk about chronic wasting disease. This is a cervid disease. It's caused by a prion protein and is 100% fatal. So an animal that's infected, if allowed to survive through the infection or live with the infection, they will eventually succumb to the disease. Transmission rate increases with population density, and this is specific to early during an outbreak. And that's because, so initially during an outbreak, this disease is transmitted directly from animal to animal. So there's no intermediate host, there's no vector. A sick animal is going to shed prions in their saliva, urine, and feces. With direct contact with susceptible animals, they can transmit that disease. So early on in an outbreak, that's typically how transmission is occurring. Now, it doesn't necessarily have to be transmitted that way. It can actually be transmitted indirectly through contaminated environments. And that indirect transmission will become much bigger of an issue as outbreaks progress through time. And that's because as sick animals shed those prion proteins in the environment, that contamination builds up, and then indirect transmission becomes more of a driver of how the disease progresses in the population. So if we just focus on early during an outbreak, when transmission is direct contact, it really is a density dependent disease, because if you have a lot of animals together and congregated and at high densities, they're going to be more efficient at transmitting CWD to one another. And that's why with this disease, we're very concerned with artificially concentrating animals. And that's why a lot of times we talk about how do we manage populations, reduce densities and reduce those transmission hotspots at like salt licks or areas where people are feeding. CWD has been shown to be associated with population declines in deer and elk. So there are 3 specific studies, one of which is one I conducted during my PhD in Wyoming. Another study looked at elk in Colorado, and they found that declines were detected after CWD was at 13% prevalence in the population. In white-tailed deer and mule deer in Wyoming, we saw annual population declines of 10 and 21% in white-tailed deer and mule deer, when prevalence was as high as 42% in white-tailed deer and 24% in mule deer. And so what's happening here and why we're seeing such major impacts of CWD on these populations is because it's reducing adult survival of infected animals. So in the study that I conducted, as well as other studies, we've shown that CWD doesn't actually affect an animal's ability to become pregnant. So we didn't see a change or reduction in pregnancy. And we also saw very minimal declines in recruitment that wouldn't suggest that that's impacting the population, as much as the fact that infected animals do not survive more than 2 years with the disease. So it's really reducing that prime age adult survival. And I think Scott touched on this a little bit, but typically in ungulate populations, adult survival is constant, with offspring or recruitment being more variable from year to year. So you have this disease now that is significantly reducing adult survival, and adult survival has the greatest impact on populations due to their contribution, I should say female adult survival, due to their contributions to population productivity. So what I mean is small changes in adult survival will have the largest effect on population growth rates. So that lambda that you heard about earlier, compared to an equivalent change in any other vital rate, so like recruitment. So that is why as managers, we're extremely concerned about CWD, because it is affecting that prime age adult survival, and it's having pretty significant impacts on populations. Okay, so that was the first factor that we'll talk about. So the next factor, so shifting gears, we'll talk about that potentially influencing populations is resource competition. So when we talk about resource competition, what we're generally talking about is how do animals acquire food, water, and shelter to sustain life and promote production or reproduction. And so what we mean is, are they able to acquire habitat? This is a density dependent factor, and I think Scott alluded to this. This is a very classic density dependent factor. So if there are fewer individuals in the population, generally resources are not limiting the population, so animals are able to acquire quality habitat. As the populations grow and with more and more individuals, resources become limited as the population approaches that carrying capacity. So either that the ECC or the NCC that Scott talked about. So competition can be intraspecific. So individuals are competing with members of the same species, or interspecific, so they're competing with other species on the landscape that share their same resources. So that's pretty straightforward of what resource competition is. So how does this actually play out in ungulate populations? So I'm going to give one example of how what this looks like in ungulate populations. So why do animals migrate? They migrate to access high quality habitat. So again, trying to access food, water, shelter. But you might know that there are some animals that don't migrate. So these animals are known as residents. And the reason why they don't migrate is that the costs of migrating outweigh the benefits. And so what that means is that their survival is actually maximized if they remain on their home range year round. So they don't use separate seasonal ranges for winter or summer. So they stay put in the same area all year round. And then there's this other phenomenon that is seen in certain populations known as partial migration. And this is where some individuals migrate while others do not in the same population. And this typically occurs when populations are experiencing density dependent factors, and where habitat is going to vary by season. So, so with this in mind, how do we test this? So how do we actually test this in populations? So we can look at, or we can test the competitive release hypothesis. So this hypothesis states that competition for a limited amount of home ranges of relatively high quality should result in residents having home ranges of higher quality than migrants in populations experiencing density dependence. So what this basically means is that if you were to look at habitat quality of animals that don't migrate, they should, in theory, have much better habitat compared to those animals that do decide to migrate, because the cost of, or I'm sorry, the benefit of migrating is that for those individuals that are not able to compete for better habitats should decide to leave those areas in search of higher quality habitats. And so there was a study that was actually conducted in Northeast Washington of white-tailed deer, specifically in Stevens County. And that population is a partially migratory population. So some whitetails will migrate to different seasonal ranges, whereas others are resident animals. And so the purpose of this study was to evaluate whether spring migration of female white-tailed deer could be predicted by winter habitat quality. So, really testing this competitive release hypothesis. So if you can evaluate the winter habitat quality of all these deer based on winter habitat quality, can you predict whether or not those animals will migrate or will they be resident? So here in this figure, what I'm showing you is how they measured habitat quality. So so they used home range size in the winter, as well as proportion of cropland within their winter home ranges. And so that's on the x-axis. And so on the y-axis, you have the probability of migrating. So if you just look at that first figure, so we have home range size on the x-axis, and it's increasing in size from left to right. And so as the home ranges became larger in the winter, the probability of that animal migrating also increased. So what this means is that so what this means is that animals that had larger home ranges in the winter, in theory, had poor quality habitat, because it's been shown that home range size is density dependent and can be used as a proxy for habitat quality. So animals have to increase their home range size in order to access all of the resources that they need. So larger home ranges mean poor quality habitat. So what this research found was that they could predict that with larger home ranges, so poor quality habitat, those animals actually do decide to migrate. And a similar relationship was shown, now if you look at the figure on the right, with cropland. So another metric or indice of habitat quality. So greater amounts of cropland within a deer's winter home range meant better quality habitat. So individuals with on the right side of that graph with more cropland within their winter home range had a lower probability of migrating, so were more likely to be a resident animal. And you can see that relationship on that figure. So in this study, they were able to predict that with lower quality habitat on winter range, those animals would decide to migrate. So what does this all mean to the actual ungulate population, or the white-tailed deer population in this case? Well, it all has to go back to survival. So these individuals are migrating to survive. So partial migration is an evolutionarily stable strategy. And so what this means is that individuals will choose to remain resident or migrate to maximize their individual fitness. And the way that this research was able to show that was that in theory, if the decision to remain resident or to migrate affects survival, we should see a stabilization in those survival rates. So migrants and residents should have pretty similar annual survival to mitigate the effects of limited resources. So, and that is what they found. So this table here shows the survival rates of those adult females in winter and summer, and comparing migrants versus residents. And you can see that those survival rates are very similar and overlap between migrants and residents, both in winter and summer. And so what that means is that those migrants were able to mitigate any potential costs of intraspecific competition or density dependence that's occurring in that population by deciding to migrate and access resources that they wouldn't have if they remained a resident on the landscape. So that is just one way, one example of how resource competition can play out in the behavior and change the population size of ungulates. Okay, so the next factor that I'll talk about is predators and how they might influence ungulate populations. Okay. I'm going to take a drink real quick. All right, so predation. Simply speaking, it is the killing of one organism by another for food. So that's very simplistic. That is the act of predation. But what is the effect? And this is where it gets a little more complicated, or I guess more interesting. So the effects of predation can work on 3 different levels. So it can work on the individual level, the population level, as well as the community level. And these all build on each other. The effect at the individual level, it can be as simple as the animal lives or dies. And then there's also another effect called indirect effects of predation, which I'll actually talk about here in a little bit and go into more detail. But so at the individual level, it seems pretty self-explanatory about what the effect of predation is on those individuals. When you move into the population level, what really matters is the predisposition of the prey of the individual. So what we're talking about is prey vulnerability to predation. And so prey vulnerability can be influenced by the prey's age, sex, nutritional condition, whether or not they are affected by a disease, the weather, their location, so wrong place, wrong time, so time of day. So certain predators are more active during certain times of the day. So all of these different categories of prey vulnerability influence how predation affects population. And that's because depending on the prey's predisposition to predation can influence whether or not predation is compensatory or additive. And so what that means is if a prey animal individual is predisposed to mortality, but it is preyed upon or killed by a predator, even though that animal was killed and taken out by a predator, if it was not, would it have survived or would it have died from some other cause? So if that animal was already predisposed to mortality, so let's say it was in poor nutritional condition, potentially if that animal does somehow escape predation, it might die because of its poor condition anyways, and be a loss for that population. And that's an example of a compensatory effect of predation. On the opposite end of that spectrum is an additive effect of predation. So that animal, the prey, is not predisposed to mortality, but it is still removed from the population by the predator. And so, so you might also hear terms like proximate versus ultimate cause of mortality. And this is getting at this predisposition component. And so while the proximate or cause of mortality is often recorded by researchers, so like we'll go out and we investigate a mortality of an ungulate and we know that it was killed by X predator. What's a lot harder though is to determine the ultimate cause of mortality. So was that prey species already predisposed to mortality, making them more vulnerable to predation? So that ultimate cause is really what we're trying to get at, but it's much harder to document and record through studies. So at the community level, community effects are highly localized and site specific. So the effects depend on multiple factors, from the individual to the population level effects, and can include things like prey disposition, where predation falls along the compensatory or additive continuum, the numbers and characteristics of the predator species, the numbers and vulnerability of alternative prey in the population, in those communities, and then also the prey population age structure could play into the community effects of predation. So additive predation can drive populations to low level densities, and in contrast, compensatory mortality tends to dampen that variability or variation in those systems and can act as a negative feedback mechanism. And that this is kind of what Scott described as a regulatory or regulation factor, and it works to keep those systems at their current equilibrium state. So, so what's important to remember is that, yes, while predation, the act of killing an organism and taking them out of the system, is easy to quantify, the effects are much more difficult and more complex. So let's talk a little bit more about this question of, is predation compensatory or additive? And again, it's going to fall along this continuum. It's not often, I should say, one or the other completely. And it depends on things like prey predisposition, the predator composition and diversity in those communities, and then also sometimes what are the alternative prey in those communities. So what this figure shows, it's a really nice depiction of how to determine whether mortality is additive or compensatory, or if it falls somewhere along that continuum. So so in this plot on the y-axis, you have survival, and on the x-axis, you have predation rate. And so if predation rate, a change in predation rate, it has an equal effect on survival, then you get this slope equal to -1, which means that that mortality source is completely additive. So if you have a 20% increase in predation rate and you see a 20% drop, so survival go from 1, you'll see survival go from 1 to 100% to 80%. That would mean that source of mortality is completely additive to the population. So on the opposite side of that spectrum, again, you have completely compensatory. So there is no relationship between predation rate and survival. So no matter where you are, whatever the predation rate is, survival is unaffected. So you have this slope equal to zero. Now, most mortality sources are going to fall between those two extremes, and you'll see slopes that either are primarily compensatory or primarily additive. And so that those slopes that are greater than -0.5 are going to be primarily compensatory sources of mortality, versus those that fall below -0.5, and those are going to be primarily additive sources of mortality. So you'll see graphs here on out that look similar to this that describe these relationships. So for example, excuse me, there was a study that was conducted that looked at, it synthesized information from 5 different Western states, and it looked at 12 different studies of neonate elk survival. And what they made a similar figure where they have cause-specific mortality rate on the y-axis and neonate survival on the, on the y-axis, and cause-specific mortality rate on the x-axis. So same setup as the figure to the left. And if we look at the slopes of the various predators in those systems, we see that the only predator, bears, had a completely additive effect on neonate survival in this study. So, and actually that slope is -1.01. So what they found was that grizzly bears, their predation occurred early. So it was likely not a function of vulnerability of the prey. So they were killing neonates during that hiding phase of their life. So really from birth to like 2 or 3 weeks out. And so their vulnerability, their predisposition probably wasn't kicking in at that point. And so so grizzly bears in particular were most likely just spatially overlapping with their prey and were opportunistically killing neonates as they found them. So there was nothing predisposing those neonates to grizzly bear predation. And so the effect of bear predation on those neonates was in this case completely additive. And when you look at the slopes of those of the other carnivore species in this system, these systems, they fall within that continuum, so not completely additive or compensatory. So bears really were the only predators of neonate calves in this study of elk in 5 Western states to be additive in those systems. So, okay. So there are also, as I alluded to earlier, indirect effects of predators. So this isn't the direct killing of prey. This would be the non-lethal or indirect risk effects of predators on prey. And so the direct effects of consuming prey are more easily observed, obviously, but the non-consumptive risk effects on prey populations tend to be more challenging to empirically measure in populations. So let's see here. So there was this really elegant study conducted in the late 90s of predatory spiders on grasshoppers. And what this study did was they actually glued the mouth parts of the spiders, some of the spiders. So those spiders actually couldn't directly kill their prey, but they were still present in those systems. And so what this study found was that the grasshoppers, the prey, did not distinguish between predation, between predator spiders that actually could kill them and what they called risk spiders, because they couldn't actually kill them, but they were still presented a risk to their prey. And so both grasshoppers exhibited significant shifts in their feeding time budgets in the presence of spiders versus when the grasshoppers were alone. So no spiders were in those systems. And so what they found was that survival was actually the same and reduced in both populations of grasshoppers, even in populations where the spiders couldn't actually physically kill the grasshoppers. So there is this non-lethal risk effect that was occurring in this system. So, so we can't obviously glue the mouth parts of carnivore species to do something like this. So it's a lot more challenging to measure the risks of predation and then the response of prey. But there are individuals and researchers trying to do this. And so what's really important when you're trying to understand predation risk and response is distinguishing between what predation risk is and what the risk effect is. So predation risk is the probability of actually being killed by a predator. And the risk effects are the costs of those anti-predator behaviors in response to risk. So those anti-predator behaviors are trying to reduce predation risk. So those two things are very different. So an example, a hypothetical example of this that Prue et al. Present in their paper is that let's say you have two different bighorn sheep populations. They both have identical probability of being killed by cougars. However, it's possible that sheep in one population might be displaying anti-predator behaviors that are more costly to those sheep. So for example, they might have higher vigilance rates. They might forage in poor habitats to avoid predators. So in this scenario, their predation risk is identical, but the risk effects are quite different in these two populations. So it's really important to distinguish between these two variables. So they developed, so these researchers developed this really nice visualization or hierarchy of how you can, different risk metrics that you can measure, as well as response metrics. And so what this shows is that the risk or the probability of being killed can be broken up into different phases: the encounter phase, the engagement phase, and the attack phase. I don't worry too much about that, but there's different levels of how predation risk can be measured. And so within those phases, there's different ways you can measure risk, and that's you can look at where predators occur on the landscape through occupancy models. You can look at kill site characteristics. They even make collars with cameras on them now. So as you go down in that list, you have stronger inference about the impacts on the prey population. So those measures of risk are better at the bottom of that list. And then if you look at the response metrics, that can also be broken down into different areas of response. So prey can respond behaviorally. They can have physical responses, physiology. So you could look at hormone levels in response to predation risk. But more importantly, and what we strive to do, is measure responses to prey demographics. So changes in those vital rates is what we try to strive for when we're measuring the risk effects of predation. So to build on that, and what makes this even more complex, is that most predators and prey do not work in systems that are these one-to-one interactions. So generally, we're looking at systems with multiple predators and potentially multiple risk effects. So this is just a visualization of how one might attempt to get at measuring these latent variables like predation risk and risk effects. And so potentially you could look at the occupancy of the different predators in those systems. You can characterize their kill site locations or look at mortality rates. And then that gives you a metric of the predation risk. And then predation risk could then be linked to risk effects through measures of risk signals. So in this picture, they show risk signals could include calls, scats, tracks, and other habitat features that can measure the response to risk. So and then for risk effects, you can look at, again, those three different categories of the response metrics, like behavioral responses, demographic response, or stress response, by measuring any one of those response metrics in that list that you see on the left there. So basically, all this to say is that there are other effects of predation, not just the direct effect of an animal killing another animal and taking it out of the population. And so what this is supposed to highlight for you is that these systems are very complex, and the way to measure the effects can be pretty complicated. And it's not, and it can be, depending on the variables that you're measuring, can really change or impact what your inference is of how predation risk and is affecting those prey populations. So, so I know that's really confusing, but it's just to highlight how complex these systems are and how careful we have to be with what types of inferences we're making based on what we're collecting from those populations. Okay, so that's all I have to say for predation. So the next factor that I'll talk about is climate. We're shifting from our density dependent factors, and now we're going to talk about a density independent factor. This is also known as an abiotic factor. So those other factors were biotic factors. Abiotic factors are non-living factors that affect populations. Here I could talk about, climatic extremes like, well, Scott gave the example of Mount St. Helens erupting. Basically any sort of natural disaster that's going to kill or directly impact a population. Those effects are pretty straightforward and really would only affect populations if those populations were small and isolated. So what I want to talk about when we're talking about climate is how climate actually interacts with other biotic or density dependent factors and how those interactions affect populations. So here I'm showing that climate can interact with forage availability and quality. It can also interact with predation rate. And then I'll also talk about how climate can interact with diseases to affect populations. So I'll go through these three interactions with some specific examples. So let's first talk about that, how climate can interact with forage to affect populations. So, excuse me. So as an example of this, there was a study conducted on moose populations, looking at moose populations in Wyoming, Colorado, and Utah. So just as a reminder, for ungulates, which are long-lived species, their life history theory predicts that nutritional reserves are allocated to reproduction in a state-dependent manner. So basically what that means is that for long-lived species like moose, they're going to conserve survival, and depending on their nutritional reserves will depend on if they reproduce or not. So additionally, as per capita food abundance and nutritional reserves decline, reproduction and recruitment become increasingly sensitive to weather. So this degree to which weather influences vital rates like recruitment should be associated with how close the population is to that nutritional carrying capacity that Scott talked about. And so this is what we call the nutritional buffer hypothesis. So so basically what this means is that, so when they looked at these moose populations and they measured calf-cow ratios of various moose populations that were either stable or declining populations, they found that that moose populations that were stable were not impacted, or not, calf-cow ratios were not influenced by the climate or the weather of from the previous year. So basically what this figure is showing is that in stable populations where presumably the moose population is well below that nutritional carrying capacity, during hot, dry, so poor growing conditions years, calf-cow ratios were similar to years where growing conditions were good, so they're cool, wet conditions in those populations. So basically, what this is saying is that climate in these populations weren't influencing reproduction in stable moose populations. But now if you look at moose populations that are declining and presumably much closer to that nutritional carrying capacity, now when you look at calf-cow ratios as a response to climate variation, so hot, dry seasons, hot, dry seasons, when you looked at calf-cow ratios the following year, were reduced compared to seasons when the population experienced really good growing conditions. So those calf-cow ratios increase. So recruitment was sensitive to climatic variations only when those moose populations were close to their nutritional carrying capacity. So this is also a reason why if you were to only look at moose populations that were not close to nutritional carrying capacity, you might inadvertently not recognize or you wouldn't observe how climate actually potentially could impact these populations, or how it interacts with the forage and how it impacts the nutritional condition of these animals. So this is just an example how climate does interact with a biotic factor, forage, to impact reproduction and population size in these populations. So now let's talk about climate and how it interacts with, potentially interacts with predation. In a study that was conducted by looking at cause-specific mortality of 45 elk populations throughout the West, and including Alberta, Canada, they found that elk populations with well-established wolves, mortality was higher in years with high winter precipitation. So in this figure, that's the darker bars. So in populations with wolf population, in elk populations with wolf populations, sympatric with wolf populations, you see that adult female elk survival is pretty constant until you get to these higher mean winter precipitation categories. So then all of a sudden wolf predation becomes, interacts with climate, creating this decline in annual survival in adult female elk. The lighter bars represent populations that don't have, that are not sympatric with wolves. And you can see that that survival in these populations is pretty constant and not dependent on mean winter precipitation. Although you'll see that in the two highest categories, they didn't have many populations without wolves that experience those more extreme winter precipitation levels. So basically what this is showing is that climate, so in this case, they measured winter severity. Will interact with how effective wolves are with predating adult female elk. So deep snow likely increase the susceptibility of these elk to predation, and that could either be through effects of nutrition. So these elk weren't in great nutritional condition because of the severe winter, or just simply their ability to escape predation. So maybe the deep snow hindered them from actually escaping predators, but they didn't measure those. So basically showing how until winter gets to a certain point, wolf predation doesn't really impact the annual survival of adult female elk. Okay, and lastly, let's talk about climate and its interaction with disease. Going back to the example of hemorrhagic disease that uses a biological vector to transmit from animal to animal, this is showing the probability distribution of Chilocoides sonorensis, which is the primary species to transmit blue tongue virus and epizootic hemorrhagic disease virus. And so these maps show under different projected climate change scenarios, the expansion of the biological vector throughout Montana and more north into Alberta. And so what this is potentially predicting is that with the expansion of the biological vector, we potentially will also see an expansion of outbreaks of blue tongue virus and EHD virus in deer populations. So this is really concerning with these projected models of climate change. And so populations that are already, dealing with other limiting factors potentially have this new threat in several decades because of this expansion in the vector and potentially these viruses. Okay, so the very last factor that I'm going to talk about is human activity or human disturbance. Human impacts can be both direct and indirect, so very similar to predators. I could have talked about humans when we're talking about predators, but while while we do take prey just like predators do, we also have these other very unique ways we interact with ungulate populations and potentially impact those populations, very unique from carnivore predators. So there are direct impacts like through harvest. We also manipulate their habitats. We create obstacles that are difficult and challenging for them to navigate. So for example, fences, so that I think that picture is showing individuals demonstrating how pronghorn typically cross fences. So in order to make fences wildlife friendly, they typically exchange that bottom strand to a smooth wire instead of barbed wire, so it's easier for them to go under the fences. We also have other indirect impacts, so like just even our presence, like with other carnivore species, there are risk effects associated with even our presence and how we use the land, either and how we recreate. We also can change or manipulate ungulate behavior that will potentially affect their populations and survival. So for example, there are certain scenarios where we feed animals to essentially stop them from going to their traditional winter ranges, to reduce conflict with people. We also do that in certain areas to mitigate issues with disease and interactions with wildlife and livestock. So there are many, many different ways that humans interact and impact ungulate populations beyond what even carnivores do to these ungulate populations. So I'll touch on just a few examples of this. Of most interest probably is harvest. Harvest tends to be an additive source of mortality. And that's because typically we remove through harvest prime aged adults. And again, going back to that prime-aged adults have the highest reproductive value in the population. And so harvest tends to remove those individuals as compared to carnivores, who typically will remove individuals that are predisposed to mortality. So the young and the senescent animals. I think that's one of your terms in the glossary, is senescent animals or older aged animals that with really low or non-existent reproductive value. So an example of this, and this was demonstrated again in this elk study, when they looked at adult female survival, elk survival, and compared that to and looked at cause-specific mortality rates of harvest, cougars, wolves, all carnivores combined, and then some unknown sources of mortality. If you look at this graph and you remember the slopes of additive versus compensatory, if you look at harvest, you see that almost perfect relationship between survival and cause-specific mortality rate on the x-axis for harvest. And so what they found was that harvest was the only factor where their cause-specific mortality analysis had an additive effect on the total adult female elk mortality. And so survival rates were substantially higher in unharvested populations. And then in elk populations that they studied, harvest induced a greater absolute change in survival than did any other source of mortality. So what's really interesting is if you look at annual survival and you break it up into populations with either no predators, populations sympatric with only cougars, and then populations sympatric with cougars and wolves. If you look at baseline survival, which means they took out in their analysis all human-induced mortality, so all harvest, survival is pretty similar among those populations. But whenever you add in harvest, in populations that had no predators or cougars only, annual survival does decline, but not greatly, and it's very similar between those two. But when you look, what's really interesting is when you look at populations total survival, when you add in harvest, where they're sympatric with cougars and wolves, you actually see an increase in adult female elk survival in those populations. So what this shows is that when harvest was added, the influence of native carnivores became compensatory. So basically what wildlife managers were doing was that they were using harvest as a management tool to offset the impacts of carnivores, especially in those systems with recolonizing wolves. So, basically what this means is that that harvest was an effective management tool to mitigate those potential effects of wolf predation in those populations. So I thought that was a really interesting outcome when they looked at these elk populations. And again, remember, this is a study that looked at several different populations throughout the West. So it's a really broad geographic scale. So on a broad geographic scale, these are the patterns that they showed. Now, if you were to do this for any specific population, these patterns might not be the same in any one population, but on a broad geographic scale throughout the West, this is what it was shown, that harvest was additive. Okay, and so my last slide, or my last example, of the indirects of human activity. I'm going to talk about the Teton bighorn sheep herd. So this herd was once migratory, but is now limited to higher elevations year round in the Teton Mountains outside of Jackson, Wyoming. And so these sheep used to migrate down to the valley bottoms in Jackson, but because of development, they now remain high in the mountains year round to eke out a living up there. And so what's really interesting about this population in response to the impacts of humans is that they have developed a alternative foraging strategy and what is called an abbreviated migration in response to the fact that they no longer can migrate down to the valley bottoms. So typically ungulates will migrate to lower elevations in the spring, to lower elevations in the winter, and then migrate to higher elevations and follow green up during the spring to summer ranges. Well, this population of bighorn sheep actually does the opposite, and they remain high in those high in the mountains. So they actually migrate to higher elevation in winter, and they do an abbreviated migration to lower elevations in the spring, just where those snowbanks are melted and it reveals green forage. And then they slowly work their way back up, following green up to their summer ranges that are just as higher or just a little bit higher than their winter ranges. So they kind of have this opposite strategy for foraging in response to the fact that they no longer can migrate down to the valley bottoms. What this has done, though, is it has isolated this bighorn sheep population to the Teton mountain range, which has had some benefits. They have essentially cut themselves off from other bighorn sheep herds that are affected by pneumonia. And so they have not experienced a pneumonia outbreak, but it has also made them incredibly susceptible to potential outbreaks or catastrophic loss because they're so isolated. They've lost a lot of genetic diversity in this herd. So in addition to the fact that they've had to completely change their foraging strategies and where they're making a living because of anthropogenic impacts, but now research has shown that on their winter ranges, they now are being they're having to avoid certain areas on their within their winter range that is suitable bighorn sheep habitat because of recreation, specifically backcountry skiers. And so what this map shows is that all those yellow lines are skiers who carried a GPS with them when they were skiing in the backcountry. And what they found was that bighorn sheep were avoiding those areas, even though there is good habitat, good winter range habitat they should be using in those areas. But unfortunately, they're avoiding them because of the human disturbance piece. So the purple splotches are where bighorn sheep were relocated. They were wearing GPS collars. The red shows potential winter range habitat. And then when they accounted for skier activity, it greatly reduces the amount of potential habitat available to those bighorn sheep. So this is just an example of how anthropogenic effects how anthropogenic impacts ungulate populations. This bighorn sheep population was resilient to the fact that they no longer could migrate, but unfortunately there are these additional human impacts that might be really disrupting this herd and potentially threatening their viability into the future. So just a really interesting story about how this bighorn sheep herd is trying to make a living in the Teton Mountain Range. Okay, so that's it for my presentation. These are the citations, some of the research that I presented examples for during the presentation. And I am ready to take questions. I will say that especially the study on the adult female elk, Scott McCorkadale was one of the co-authors, so I'm sure he can help with answering a lot of questions. And yeah, that's it for the presentation.